This method can help answer key questions on protein localization and dynamics in photosynthetic organisms.
By combining the photobleaching method with super-resolution microscope, we are able to detect the protein dynamics of photosynthetic organisms in a remarkable detail. This method not only provides insight into the protein dynamics of Prochlorococcus, it can also be applied to many other organisms containing radiorhodopsin and bacterial chlorophyll.
Demonstrating this procedure will be Yaxin Liu, a graduate student from my laboratory.
To begin, add 5 milliliters of seawater-based Pro 99 medium to a culture flask and inoculate it with 1 milliliter of Prochlorococcus Med4. Grow the Prochlorococcus at 23 degrees Celsius under a light with an intensity of 35 micromole photons per square meter. After 5 days, collect 1 milliliter of the culture into a 1.5 milliliter tube. Add 100 microliters of freshly prepared formaldehyde to the tube to fix the culture. Incubate in the dark at room temperature for 20 minutes. Then spin down the sample at 13,500 times G for 1 minute. Remove the supernatant and resuspend the cells in 100 microliters of Pro 99 medium. Store the sample at 4 degrees Celsius in the dark until ready to perform immunostaining. First, vortex the vial of polystyrene beads. Using 50% ethanol, prepare a 1 to 20,000 dilution as a working slurry. Turn on the hot plate and set